And for the first 2 or 3 years, that's what the company did. The whole array of different proteins where they made molecules to those proteins and showed that they could actually get them degraded. So very, very exciting. And certainly when I joined the company, the stage where they were at was working particularly on two programs, the androgen receptor and the estrogen receptor, one for prostate cancer and one for breast cancer. And they were probably thinking they were maybe 2 years away from a potential clinical candidate. And I think by me joining, bringing some big pharma kind of thinking about, when is it good enough to move, we're able to move those programs forward relatively quickly and get them into the clinic within that 1.5, 2 years timeline. And that was the first time really getting these new model compounds into humans and showing that the preclinical data that we'd seen actually could translate into the clinic. And really the exciting part was everything we were doing was the first time it was being done. It's the first molecules made, the first ones that got to the healthcare, the health authorities approved to get into humans, the first clinical data, which was incredible data. And it was exciting just because people thought maybe these PROTACs wouldn't act like normal small molecules because they're larger than your average small molecule. And so they would have different properties. And so it was very exciting to see PK, PD data in the first initial number of patients. And of course, since then, moving on to seeing efficacy data in a number of different programs as well. So those initial days were very exciting just because it was a first of every single time we were working on it. And now protein degradation as an area has grown quite a bit. There's a number of small companies, probably about 60 or 70, at the last count, they're in the protein degradation space in general. And of course, the big pharma companies have protein degradation as one of their, I'd say, normal modalities now. So it really, over the last 8 years, 10 years, has really migrated into being another potential therapeutic approach that small and big companies take. And our challenge has been to stay as best we can in the lead in terms of innovation and moving our portfolio forward.
Yeah, absolutely. So whenever you look at the things that you're working on at Arvinas, what are some of the really strong breakthroughs that you're most proud of?
Yeah, so one of the clear initial breakthroughs was going back to this issue that PROTACs are, larger small molecules, or they can range from 8 to well over 1,000 kilodaltons. So they don't naturally lend themselves to drug-like properties, the ones you'd like to see for a drug, in other words, the oral bioavailability. So the company's focus, the chemist's focus for the first few years was trying to find those kind of properties that could be imbued into a PROTAC that would make them more drug-like, ideally make them orally bioavailable. And the chemists largely did this by ignoring the kind of classic rule of 5, really trying to find a different set of rules for PROTACs, using biology in a different way, doing a lot more in vivo work well up front to see signs that could give chemists a lead into saying this particular ligand, this particular linker actually gives potential oral bioavailability. And over a few years, we cracked that as an issue to really show that PROTACs could be made readily orally bioavailable. And almost all the programs that we work on now have PROTACs that are orally bioavailable. And then we're able to segue that into understanding how to make them brain penetrant. And once we did that, that opened up the whole area of neuroscience, and particularly neurodegeneration, to us in terms of our portfolio. So those are the significant things. Taking that initial technology platform that Craig had basically invented and handed to us, and then adding different features like oral bioavailability, brain penetrance, understanding PK/PD relationships, then an array of different programs where you can see the proteins being targeted highly selectively for protein degradation. So a number of really quite significant firsts moving the technology forward. And of course, that has helped the rest of this kind of mini industry in protein degradation. So a lot of companies obviously know now how to make them orally bioavailable, and they're learning how to make them brain penetrant. And that's just added to the fact that the technology is, obviously ready for prime time as a therapeutic.
So I also read that you also have collaborations with other pharmaceutical companies. How have those partnerships contributed to your mission?
Yeah, great question. And when I joined, the company had already had a couple of deals done right at the very early beginnings of the company's history. A target a deal with Merck, one with Genentech, and then when I joined, the Genentech one was expanded. We also did a deal with Pfizer on a target deal, and these were the companies handing us their interesting targets. We would find degraders and hand them back to them. So we weren't getting them into our portfolio, but they were going into those companies' portfolios. And the last deal we did of that type was with Bayer. And we also, at the same time, with Bayer Agrichem, set up a joint venture in the agrichem space. We made a company called Earth Bio, based in North Carolina, and we imported all of our technologies. And they've been focused on looking at how to deploy that technology into understanding disease control in plants and insect control. We're basically showing you the ubiquitous nature of protein degradation mechanisms. And plants have many, many ligases, many more than humans. But the basic technology of making heterobifunctional molecules works in plants just as well as it does in humans. So yeah, there's been a kind of an interesting kind of migration of the technology over the last several years.
Very good. Can you share a little bit more, a little bit about your experience with both large pharmaceutical companies and smaller biotech firms? And what do you see as the key differences and challenges in leading these different types of organizations?
Yeah, so when we did the first deals, it was a great validation for the technology. So for any small biotech company, getting a deal done with a Merck or a Pfizer is just huge validation for the technology. You also get access to really smart scientists in the pharma company, and quite a lot of time you're getting access to their capabilities. So that helped Arvinas grow as we were working for these companies to find degraders for them. We were also learning and getting access and capabilities, which was incredibly helpful. And then we did a really significant deal with Pfizer on our major asset, vepdegestrant, where we did a 50-50 co-development, co-commercialization deal. And that is a very different, very, very different beast altogether. This is a co-sharing development, and for the last several years, where teams are working very closely together, sharing common tools, sharing common vision, really moving an asset forward. And what you're dealing with there is we're a small company working with a very, very large company. So things might change for us, but probably don't change as much as they do for Pfizer. So that's one of the challenges with a big pharma company. They do have this global footprint. They have many, many things going on simultaneously. And even though we've got this one very exciting asset, they have many. So keeping that intensity as being part of the kind of, I think one of the great things that Pfizer has done, keeping that focus on vepdegestrant and moving the asset forward, even though I know they've got many, many other things they could be doing. But it's worked very, very well. But it's a different scenario from just working on your own stuff. You're in those other deals, you were working for those companies to give them PROTACs that goes into their pipeline. And now you're in a co-development where you have to share and make joint decisions. But it's been a great learning experience. And we wouldn't have been able to develop vepdegestrant to the stage it's at, literally just waiting for pivotal data in the next few weeks and months without Pfizer.
Yeah, I'm less familiar with, I would say, the drug development collaborations that happen quite frequently, but much more familiar with collaborations for building things like personalized diagnostics and things like that in life sciences. And I always know that we would typically show up on calls and it'd be this level of anxiety because essentially you got the big brother and their smart scientists looking back at the things that you're doing, and there were always questions that they would oftentimes ask that, frankly would carry us much further as well. I mean, so it does provide great benefits, and, I could see that as well.
Yeah.
Yeah, for sure.